Millán, that is going to explain now using network community clustering algorithms to aid determination of protein structures. Please give her a welcome applause.
Well, good afternoon, everyone, and thanks, first of all, to the organizers for giving me the opportunity of talking to you about some of the work I do, and also to you for attending to this talk. So, first of all, let me introduce myself. I'm a PhD student at the Structural Biology Unit in the Archimboldo team in the crystallographic methods group. And before I ended up there doing my PhD, I studied a 5-year degree in biotechnology and did a master's in crystallography. In these 5 years, I have learned all I know about programming, which is a little bit, and with much great help of my colleague Massimo, who comes from a background in computer science. So in this sense, this synergy that has been mentioned in other talks has been really good. And well, I have some knowledge of other languages because of work during my degree or some small projects. But basically, all the software we develop is in Python, and Python is the tool we use for our daily work. So just a brief outline of what I'm going to speak about. So first, we'll talk about proteins and their structure. Then about how do we get this protein structure, so which is the actual experimental method, because we still need experimental methods to get them, and how Python is helping us in this task. So this is the third talk today about proteins, so maybe I don't have to go so much into the details, but proteins in a nutshell, right? So proteins are relevant. I think they have already convinced you because they are basically the workers in the factory that our cells are. So they perform most of the functions that are required. They transport molecules, they respond to stimuli, they are the workers in a factory, right? And what they are composed of, they are they are chains of amino acids. A good analogy that is commonly used is that they are like a necklace with different shape beads, and these different shapes translated to amino acid means that they have different size, different chemical properties, and that will interact differently with each other. The actual sequence of amino acids that you have in a chain comes from the gene that codifies for that protein. So there is a relation by the genetic code that relates our gene with our protein. But our protein will not stay in this like linear unfolded shape, but rather the amino acids will start interacting with each other and will form this kind of a structure. So the first level will be what we call secondary structure. And this is also, I want to make a point, we structural biologists are very visual, and we love to see pretty pictures of the things. So you will usually see alpha helices in this cartoon representation and beta strands like this. But the actual atomic view of this is more like this, and here you have the interactions that are forming. Also, I think it's important to point out that the beta strands are not usually found, well, they are not found alone, but they are always establishing this type of interaction with another one, either in a parallel or anti-parallel fashion. But this is not the only level of structure. Structure gets more complicated and more intricate, and we have what we call tertiary structure, and this is for a single chain, but if more than one protein chain is needed to perform the function, we have what we call quaternary structure. So why do we care about the 3D structure of proteins? As Massimo has mentioned before, actually the 3D structure of the protein is basically what allows the protein to perform its function. And I think it's good that we go to a specific example because it's easier to understand it. So this is the structure of a protein we solved a couple of years ago. So this is from another group in our department that does the biological part. And this is from a pathogenic bacteria that causes gingivitis and other inflammatory diseases. And because our collaborators determined with our help this structure and also the structure of related proteins, they did understand how the actual mechanism of reaction work. And now they know how to tackle this pathogenic bacteria. So it has really a real-life application. So I said that we have already, Massimo mentioned that we have already information on the protein structures we know up to date. So we have a database which is called the protein data bank, and there is where we store these macromolecular structures that we have already determined by different methods, and we will see one of them on this talk. And though now we are changing to another format that supposedly will be the standard, until now the common way in which we have saved this information is these plain text files that contain information about the 3D coordinates in the space. All these databases can be accessed in a programmatic way, which is really simple, and you can retrieve not only information about the coordinates, but more information that allows you to do data mining, like about the experiment and so on. So all of this is really easily accessible. And one of the tools in Python that you can use to process PDB files is this library, BioPython, which is a general bioinformatics library, so it can do much more than reading PDB files. But one of its possibilities is dealing with this kind of files, and I'll show you a small example. This protein, if you've been in the previous talk, you already know, is hemoglobin, the protein that carries oxygen in our cells, and its structure is composed by 4 copies of the same protein. So what we are doing here in the code part, we are just opening normally as a file object in Python this PDB, and then we are instantiating the parser class from this BioPDB module. We are obtaining the structure with this getStructure method and saving this into this kind of structure object. And here I brought you 2 ways of accessing the information then. It's saved in a hierarchical way. So as we see here, we have here the whole model with the 4 chains. Then we have 1 chain. Then we have 1 amino acid. And for example, in this case, I'm interested in getting the carbon alphas, which are one of these atoms that all the amino acids have, and that form part of the chain. So I can iterate over each of the levels of the structure, the models. Some experimental methods produce more than 1 model, and that's why you have this level here. Then on the chains, and then checking in each of the amino acids if this atom is there, and saving it to this list. There is an easier way with 2 lines to solve this problem, but in this way you could see the structure. So I mentioned that we need experimental methods to obtain structure. So there is a relation between the sequence of amino acids and the final structure, but we still didn't figure out how to solve it, and we need experiments to determine this. And there are a few approaches to do this, but the one in which we work is with X-ray diffraction. So the issue with proteins is that they are quite small, and in order to be able to see them, we cannot use just visible light and look them through a microscope, but what we need is a wavelength that is small, also very short wavelength, like X-rays. However, there's a problem. X-rays are interacting, basically they are being absorbed. You can image with X-rays, so we need to exploit another phenomena, which is X-ray diffraction. And I brought you here this small GIF animation, which is the Young experiment, which proved that the light was having also this wave behavior and not only particles. So what you see here is that we have this slit here, this small slit, and that light is passing through here. If it will be just particles, it will be hitting here, and you will see just a single point in this pattern, but you see like some diffuse scattering here. When we have here another 2 slits which are separated by a certain distance, what we get is like a discrete pattern. And there is a relation between this and this. So these interferences between waves are producing something here that we can interpret and go out to this point. And we can use this with proteins. But the experimental setup looks a little bit different. So first of all, we need super bright X-rays, so they have to be really strong and focused, and in order to do that, we need to go to particle accelerators. So here we have the ALBA synchrotron, which is the main source from which we get these X-rays. And we don't just hit our protein with X-rays directly, but rather we use a protein crystal. And a protein crystal is not very different from a mineral crystal, so it's some super well-ordered arrangement of proteins that you get. And we do this because we need like the amplification effect of this. So instead of having 1 protein and get some diffuse scattering, by having a lot of them, we get this constructive interference that will be useful for us. Right, so then we hit our protein with the X-ray, and we detect the diffracted X-rays here. Right, but X-rays, in the same way that light, have this wave behavior, so they not only have an intensity, but they have a phase associated. This is like the angle in which this radiation is coming. And we lose these phases. We cannot measure them experimentally. We only have the amplitudes. And in order to go and get our electron density map and our protein structure, we do need the phases. We need to retrieve them somehow in order to be able to compute our electron density map. So how do we do it? There are, again, different ways to solve this phase problem, which is the bottleneck in crystallography. And one of them is what it's called molecular replacement. So, all right, we have already a database with quite a huge number of structures that have been solved. And we have the amplitudes from our data, we have the intensities, but we have lost the phases. Maybe we can use the phases from a structure that is similar to ours and compute our electron density map, because when we have coordinates, we can compute amplitudes and phases. There's a relation, a mathematical relation that we can use. However, we need to solve two obstacles before. First of all, we need to find a similar structure. And in order to do this, we can use the sequence, because normally you know the sequence of the protein that you have crystallized, and search in the database if there is something similar in terms of the amino acids. But then we also need to find how this model is oriented in our crystal, because this structure that you are using as a model was obtained in a different way, and you need to put it in your setup. So how do we find the orientation of our model? Well, most of the modern molecular replacement programs are based on something that some of you might know in another context, which is the principle of maximum likelihood, which in a sentence basically says that the best model is the one that is most consistent with your data. But how this translates to our case, I'm going to show you here with this simplified version of the Bayes theorem of the posterior probability. So, okay, we are interested in getting our posterior probability. So, which is the probability of our model, of our structure, given our data. This is what we get in the synchrotron, this diffraction pattern. Right, so this will be equal to the likelihood, so the probability of that our data has been produced by this model, and we need to take into account the prior probability. The prior probability of the model itself is just like the distances between the atoms that it should have, but this is similar, this is equal for all the models, so you don't need to worry about this. You don't need to take into account. Also, the probability of the data, in the case of our experiment, is also always constant. That means that the posterior probability is proportional to the likelihood. So we can use this likelihood as a target for doing our molecular replacement. So at the end, what is molecular replacement? It's just trying in a set of possible orientations and different rotations and translations to place our similar structure in our data, and the one that gives the maximum likelihood is the one that we take as the solution. This is more or less how it works, if it works. One of the issues is that in molecular replacement, one of the most important parameters that you need to take into account is the similarity between your protein and, well, your unknown protein and the model that you are using. And the way in which we measure this is with the root mean square deviation. Can I get some water? Thank you. Cool. So I mentioned, the root mean square deviation, again, is a concept that you might have heard before, but applied to protein structure. It's just an averaging of distances. So in this case here I have two alpha helices, and I have plotted the distance between the C alpha carbons, and then I use these distances here for this averaging, and this is how you compute the RMSE. So here I brought you a picture in which you have on the x-axis the sequence identity between two proteins, and in the y-axis the RMSE that they have between them. So what you can see is that, like, from 30, 40 up to 100, the correlation is good, and also the distribution is like very tight. But when you go 30 or below, you have worse RMSE, but also your distribution doesn't look, it's much wider. This is what it's called usually the twilight zone, because it's this part in which the correlation between sequence identity and RMSE is not so good. And because you need to set the RMSE, when you have models that are in this twilight zone, you need to take this into account. So how does this look like in structures? No? So I mentioned the RMSE. How does it look in reality? So I have here in purple this structure that we solve, and some of the related structure of the homologs that were available to use. So in this first one, you have 46% sequence identity, and you see that the RMSD, this value 0.7, this is quite low and quite good. This would possibly work with molecular replacement. And you see that the superimposition between them is quite good. If you go to these other three structures that are on this twilight zone threshold, you see more variety. So you have, for example, this one that is 26%, and the RMSE increases a little bit. But you go down to 15% sequence identity, which is quite low sequence identity, and still the structure is quite conserved. Or sometimes you have things like this. So you have 20% sequence identity, which is low, but the RMSE is quite terrible. So if you will be to use this model, it will not be easy to use it for molecular replacement, especially not only because there are some parts that are in different ways, but because even if the overall fold is similar, there might be like a slight adjustment that you will need to do, for example, between the beta strand in the middle and the helices on the side. So this will be a challenging kind of model to use. And sometimes the model is working for molecular replacement, actually. And what we do in the group is to develop Python programs that perform kind of, we call it non-standard molecular replacement. So we don't use this kind of full models, but other types of things. We use what we call generally small fragments. So we use these secondary structures like alpha helices or a few beta strands, and we try in the same way that Arcimboldo, the Italian painter, did the portraits like by using common objects and got the portrait. We do like the same but with the proteins in a way. So we have a number of programs. We have Arcimboldo Lite, that is the one that use alpha helices, ideal alpha helices. We have Arcimboldo Borges, which use like libraries of folds that are obtained by looking at these characteristic vectors that Massimo was mentioned in the previous talk. And one of the options is, okay, if I don't have a model that is very good, but still I have a distant homolog, so something that has not very good sequence identity, but the fold must be there, we can use it as a starting point to derive fragments. And one of the things we can do is to use our program Arcimboldo Shredder. And what we can do is, instead of using the whole model, cut it like into compact pieces. So we do like spatial partitioning of the initial model. And then identifying groups that we can like refine against each other, like do like rigid body refinement between them in our molecular replacement. So how we can do this partitioning, or why do we want to do this partitioning? So here I brought an example with a template. So this has been trim of whatever is not secondary structure, so alpha helices and beta strands. And if I don't define any group, I have here the analogy with the square will be like our final structure we want to get, and the triangles are our models. Well, if I don't define any group, I cannot fit this anything better than this. I have to worry about the rotational translation, just these parameters, but I cannot do anything more. If I then split it into three groups, for example, the helices in one side, the other helices in the other side, and the beta strands, I have more parameters now to worry about, but I can adjust a little bit better. If I then leave each of the helices separated, now I went up to 21 parameters for each of the rotation, translation, and the observation, but even if it's to a part of the structure, I can adjust my better. So in a way, I need to find an agreement of not going really to individual atoms or residues, but making groups that I can use for giving the degrees of freedom to the model. So more or less, the process that this program does, this Arcimboldo Shredder, is just taking this not so good model, trim it to secondary structure, annotate groups, and this is where community clustering will make its appearance, and then generate these like compact models that we will use for molecular replacement. Right. So again, if you've been in the previous talk, we have a way to represent protein structures as graphs or networks. And in our case, the graph that we use to represent it is a complete undirected graph. And what we have in the vertices is just each secondary structure element, so each of these beta strands or alpha helices. And then the edges have a weight that is proportional to the distance, and it can have more things into account, but the basic feature is taking into account the distance. And there's like two different questions that we want to ask to answer. So what Massimo has been talking about, which is more like the prediction or the understanding of the structure through these small blocks to look at in the PDB database. But in my case, what I'm interested in to see which is the best automatic way in which we can like break down these models and help in solving the structures that we still don't know and for which we don't have good models to start with. So the tool that I'm using to do the community clustering and finding this group is Python iGraph. And Python iGraph, in order to perform community clustering, which in a sentence is basically searching for this group of nodes that are more connected between them than with the rest of the network, it has a set of different algorithms, some of them based on different strategies. There are these face greedy agent vectors and community multi-level that are based on the modularity, which is this property of being more connected. There are some of them that support directed graphs. So this is not our case, but for some of you, if you are interested, it supports these two algorithms, InfoMap and each between us. And there are other algorithms like this label propagation, world trap community, spingless, and really depending on what you are doing, you might be interested in using one or another. So here a little bit of code. This is the function we use to call these methods for our graphs. So we need just to give us input the graph object with our structure. And directly, these community methods are methods of these graph objects. Most of them just need the weight as a parameter, but some of them can be more fine-tuned with the other parameters. And there are 3 of them, actually, that do not return directly the clustering, but return rather a dendrogram. So you can decide at which, because their approach is hierarchical, and you can decide which level you want to take. So this is the case for fast greedy, walk trap, and the edge between us. So again, depending on your approach, you might need one or the other. And the kind of things that we want that community clustering does with our structure is, and the things we want to reinforce when we do the community clustering and when we define the graphs is, first of all, that beta strands are reinforced to be in the same group, because this is like what nature does with them, so they form interactions between them, and they usually don't move a lot. They move more related to the other elements. Then, because of the same reason, we don't want that a single beta strand is in a group, because that doesn't make sense. And we want things that are more or less compact in space and that could match things that will move in reality. So we have applied different approaches depending on the type of structure that we were working with. So for the case of structures that are all alpha helical, we have used this fast greedy algorithm, which was the first one I showed. And this is the kind of groupings we've been getting, which match what it makes sense for refinement. And one of the things is that we do this refinement like iteratively. We do it a couple of times. And at the moment, what we do after this first grouping is let each of the helices to refine individually. But we think it will be better to get to use this dendrogram. I say this method returns, and then putting in a different level so that we get, for example, another level of groups that is not so big. I mean, in the case that here I leave each of the helices separated, it's a lot of groups. So maybe computational is not the best idea, and we are exploring that. And in the case of alpha-beta structures, we make a mixture of the different algorithms, and we do some combinations. So what we do is first, we use the info map using a version of our graph that has been filtered and that only contains the beta strands, and that uses a distance threshold of what we know is more or less sensible for beta strands. Then we use the fast greedy on the filter graph only with the alpha helices, and then we merge the results. This has an obvious consequence, that is that you will not have groups with alpha helices and beta strands merged. But depending on your fault, this could be what you want. So for example, in this first case and this second one, which are these kind of sandwiches where you have the beta strand in the middle and helices on the sides. Here you also have like 2 betas here. This makes sense. And in this case, we have like more like 5 repeats of helices and beta strands. And here, as I said, maybe we could put another level in which we join helices and beta strands. But still, this has produced quite good results at the moment. And this is the code for this 2 steps clustering. It was just to show you, again, the options that you have in Python iGraph to select by some of the properties of the vertices, like in this case, whether they are beta strands or alpha helices. And yeah, well, as I say, what we do is just filter, use the first algorithm, filter, use the second one, and then merge the results in a dictionary. But I guess the point is, well, was this useful? So this approach has been helping us to solve new structures. Well, the answer is yes. So this program I mentioned, Archimboldo Shredder, is using these groups that we get from the community clustering, and is using them to improve both at the rotation step and at the translation step models. And with this help, with this refinement of rigid body groups, we've been able to solve a set of not only tested structures, but also unknown structures. And here I brought you an example of how this input-output of the refinement looks like. So these were models from myosin, which is one of the proteins that carries things in your muscles. And it's an alpha helical protein. This is one of the models. So the starting model is in blue. And the model that has been subject to this refinement has changed and has become 0.47 angstrom closer to the final solution of the structure. This is a lot in terms of phasing or not phasing a structure. If you were starting at 1.2 and you went down to 0.7, it probably means that you will phase your structure. So it's quite good in terms of the result. What? The groups are corresponding. I can show you because it's actually this kind of fold. Yeah. So the groups will be probably, and this is only a part of the model, so probably there will be 2 groups in there. Yeah. So, well, in conclusion, what we are doing is using community clustering as a way to automatically determine which are these best rigid body groups in order to help us determine the structures of unknown proteins. And, well, we have some promising results already with this naive description that we have at the moment, but we could include more things rather than just distance as a weight, and or different sub-steps for deciding the groups for the refinement. And we need to try it on more structures. And actually, it will be maybe useful using this powerful tool that is describing the protein as a graph to use when we have more than one, like, say, bad model, more than one distant homolog model, try to extract information of a few of them and try to determine which are the best part of the structure that we can use, for example, or how to merge them into a single model or make an hybrid model. So this could be useful with this graph description. And, well, last but not least, I would like to acknowledge well, my group, the Archimboldo team, especially my supervisor, Isabel Luzon, and my colleague, Massimo Samito, because thanks to him, I have learned a lot. Also our collaborators in Cambridge, in the group of Randy Reed, and in Göttingen, in Germany, in the group of George Sheldig. Then to the institutions, and especially the taxpayers that pay the institutions that have supported my work. And I also want to mention, because this is a Python conference, that I'm really glad and I'm really thankful to the Python community, which I knew offline online for a lot of time, but knowing them now face-to-face is really great, and I've been learning a lot with this conference. And well, I want also to thank you for your attention. If you have any questions now about the work, please feel free to ask them. But also, if you want to contact me later on, and so I will be really happy to answer your questions.
Questions? Okay, I would like to ask about the fast grady algorithm. I mean, how it works, or which are the advantages of other algorithms? I don't know.
I can say we have following like an empirical approach here. So we have tried all 8 of them, and the one that worked better is the one we're using. But you can read a little bit about the literature of each of them, and so the ones that do like more like in the case of fast grady hierarchical approach and start like by making groups and merging them are working quite good in our case. Some of the other ones, if you don't filter, if you don't put, I'll say, much more information, if you just with the distance, they are not working so good. But really depends on the case. Actually, one of the things I show that is in the combined use of, for the structures with alpha and betas. In principle, InfoMap should be really useful and powerful when you have directed graphs, and that's not our case. But in the structures we have tests was working well with our undirected graphs and producing the results. So.
Thanks.
Okay, so I got lost in one part. So I understand that in the experiment to measure the structure of the proteins, so you got the amplitude, you didn't get the phases, and you needed the phases.
Yes.
So what I got lost is, how come you actually have a database of other proteins with their amplitude and their phases? Is it because you measure them with better stuff, or because you calculate them in? I didn't get how.
Yeah. Okay, so let me go back to that. Well, in the database, what we have is the coordinates. We have the models of the proteins. But, so let me just go back to the.
Yes.
Right. The thing is that once you know already the coordinates of the structure, let me go, yeah. You can compute what will be the, how to say, is a representation, a reciprocal representation of this model.
Yes. But how come you do have the amplitude and phases for some proteins and for others don't? Because
yeah.
How come? How do you measure?
Because we solve them first.
But like model, like a mathematical way of solving
no,
it, or you
also
measure
experimentally.
Okay,
there are
okay.
either with molecular replacement or with other technique, with magnetic resonance, or with now with cryo-electron microscopy, and also in crystallography. The molecular replacement is not the only way to get these phases in X-ray diffraction experiments. There are some experimental methods, like you can put atoms that diffract a lot, heavy atoms, and measure them. So there are other.
And those methodologies are much more. They're much harder to get, or they're
experimentally.
much more
For example, the experimental phase in X-ray diffraction means that you need to derivatize your protein and add something there that
so
will.
This way is much faster and so it improves the performance. Okay.
And you are exploiting information that is already there. You have.
So remember when I show you the Nobel Prize, Max Perutz, for the hemoglobin? That was because in that period, actually, it was so difficult to solve a structure that if you solve a structure, you got the Nobel Prize.
Oh, you
and that now there are Nobel Prize for structure, but it's for much more bigger structure, so enzymes, things that are big. No, so the things they did it with this technique. So Max Perutz did it actually through experimental phasing in crystallography, but it was hard because you don't know at the time you don't know anything. Now we have a database of all the structure. You can use this technique, and you can say, okay, my structure is similar, so I can use the phases from that. But at the time you didn't have anything. But the thing is that even if you have sometimes structures that are similar, if they are not very similar, then it doesn't work. And Claudia, with this application, allows you to extend the probability to solve, even if in cases when it's not so easy. So
Yeah, the issue is that I know that visually all of this looks really similar, but in order for the molecular replacement to succeed, it must be really, really equivalent. So even these kind of deviations will make it fail.
There's one part that I missed here. In a single piece that you cut off at some point later, can you still deform that just slightly, or does it remain a rigid body?
No. Yeah, so this
Maybe you said that. It's just,
yeah, I think
must have
maybe
missed
it's clearer in this one. So I used the full thing, like the whole model, to do the community clustering and get the groups. But then once I cut these models, each of these different color thing will be refined as a different group. So I will perform, like in this case, like 3 molecular replacement with 3 different models, all of them annotated in order to do this rigid body refine on them.
But in each group, it's a rigid body. The
Yes.
atoms do not move with respect to the other atoms in the same group.
Exactly.
Okay.
Yeah. And I never have single atoms in any group. I mean, always there are one secondary structural element at least. Mhm.
Hello. Can you go back, please, to the previous slide, I think, because where the resolution of the method is displayed. You have like angstroms underneath.
Which one?
There are like 4 type of groupings, and you are comparing how much accurate you get with the angstroms. It's just a
Yes.
technical question because
Yes.
this one, yeah.
Yeah.
If I am correct, the resolution of X-ray crystallography is around 1 and 2 angstroms, right?
See?
If you
Yeah.
are lucky. And
Quite lucky, actually. More like around 2,
or
but
quite good at it. So how is meaningful that we get to under 1 angstroms of closeness in the RMSD there? Because actually you don't get any better experimentally, right?
well, yeah, but we, yeah, I understand. But what you care is in order to get phases that are good enough approximation to solve your structure, and the molecular replacement is really sensitive to that this is really close.
But that model you are using for molecular replacement was obtained with that resolution of 1 angstrom, more or less. So I don't
Depends
know.
on the model. Yeah, depends
Well,
on the model that
maybe
you use.
at the best. So
Yeah,
But the resolution is local on every atom.
exactly.
yes, but I don't get how you can benefit from having less than 1 angstrom resolution if the experimental information doesn't get any better than that. Yeah. Maybe because it should be good enough with 1 angstrom, which is the best available experimentally.
But that, yeah, this is kind of tricky too. Well, yes, but at the end, I mean, independently of which experimental resolution you had, at the end you have a model with coordinates in 3D space, and you assign, there are some kind of factors in which we put these errors, for example, the B factors in the case of PDB structures, but you at the end have an average coordinate, so at the end, I don't know how to.
Maybe we can discuss it later because this is not Python actually. So
Okay.
sorry about that question. Anyway.
No, but
Thanks.
it's interesting.
Hello, just a small technical question. How did you did you define the graphs from the protein? I guess that every node is a residue or an atom,
No.
maybe. Maybe I missed that. I don't know if you explained
yeah,
that.
it's I went a little bit quickly because the previous talk by Massimo was explaining this, but in my
Okay,
case,
I wasn't.
in his case, they were representing vectors. In my case, each of the vertices are representing one single secondary structural element. So one beta strand or one alpha helix or each of these elements here.
Okay.
And then the edges are the distances, when proportional to the distances. Yeah?
Yeah.
Okay.
Hi. So I'd like you to comment a little bit what your experience with biopython.pdb has been, because I've doing like system like I've been doing large-scale structural analysis, and I've trying to use these to parse pretty much the entire PDB, and I've run into some trouble because a lot of the numbering in the residues in the structure is messed up and this kind of thing. So what's your experience with that?
I think that doesn't depend on the tool that you use to parse it, because this is a problem of the actual format and of the scientists using the format and not respecting the standard. And this, independently of whether you develop your own parser of PDB, or you use BioPython, or you use another one, you will find these things. And they have different options for handling with problems, and some of them may be more robust or less. So I don't know, because I don't know so much large-scale analysis, but Massimo has been using BioPython for reading all the PDB, and it's been, yeah, the MMC. But I have still to see the scientists moving to MMC at this not giving problems, and also because all the, well, you know, if you've been doing a structural analysis, but there are so many other tools related also with the visualization of this structure, or with doing some things with different tools that will need to adapt to this new format, and it will be, I think, step by step.
Or you can call the API.
Yes. Yeah.
So you can stream the coordinates, and then let PDB at the end of the site, whatever.
Yeah, actually, this API, I think it depends on what you want to get, but for many of the things that otherwise you will have to do, like grepes and text mining of the header of the PDB files, you can really use the API from the PDB in Europe, which is really, really useful.
Okay, that was great advice, actually. Thanks.
Okay, well, thanks. Now I would just want to mention that we have the coffee break, and afterwards we will do a group photo on the stage. So please be... Thanks.